What we're going to do here is we're going to try keep it under 10 minutes, and we're going to look at a blood smear, a histological blood smear, where we're going to look at a range of cells that we find circulating within our blood. The first cell, and probably one of the most important cells, is the red blood cell. So the red blood cell, or the erythrocyte, is a major component of the blood. These guys contain no nucleus. When they're developing or in the developing stages of the cell, they actually chuck out the nucleus within the cell to make room for hemoglobin. And as you know, hemoglobin is a major component within red blood cells that binds up oxygen and travels around in the blood and releases it to our tissues. So one very distinguishing trait of red blood cells is that they're everywhere in the blood, for one, so they make up basically a lot of the surface area of a blood smear. They contain no nucleus. Then they're almost like this donut shaped within the blood, so you can't miss them. You'll spot them straight away. They're in your face, and you essentially can't not go past them. So the next thing we're going to look at is another major part of the blood, which is known as thrombocytes. So you'll notice in between the red blood cells there are these small buds of cells within this blood smear. So thrombocytes, or more commonly known as platelets, contain no nucleus, and these are the guys that react with coagulation factors within the blood that form blood clots. So when you scrape your knee or you cut yourself, these are the guys that kind of aggregate together to form a blood clot to stop the bleeding from happening. And essentially all they are is just fragments of cells derived from a very big cell called a megakaryocyte. And essentially this cell buds off, found in the bone marrow, and buds off these little guys into the blood. And another interesting thing is that warfarin, a drug given usually to elderly people, kind of keeps these guys from aggregating so much together to therefore reduce the chances of a blood clot forming, which could potentially be within the brain or within the heart that we don't want. So they're red blood cells and thrombocytes. What we're going to move on to now is the neutrophil. So I've circled one here. A neutrophil is one of the white blood cells that makes up the immune system. They're about 40 to 75% of all the white blood cells found within the blood, and they're part of our innate immune system, very short-lived cells and fast-acting cells in terms of response to toxic substances or injury. And they form the first part of the polymorphonuclear family, which is essentially, if we zoom in here on this particular one that I've circled, these guys have a nucleus, but it's divided into many sections. So this is referred to as polymorphonuclear cells, and it's one of the three cells you'll find in blood. They're granulated, so we can see these pink pigments within the cell that kind of give it away. So the main thing is when you're looking at a blood smear, you're going to find that they look all very similar. So you have to keep in mind that when you do the stain, it's as long as you've done it correctly, you're going to find the reference cell, and they're all going to look very similar from that. So this looks very similar, very similar stain, granulated, the multinucleus. If we go through a wander, you might be able to find some more, surely. So there's another one over here. This guy over here, just quickly, is known as a B memory or T memory cell. I can't tell the difference if it's a B or T, but essentially a B or T memory cell, these guys are part of the adaptive immune system, and they keep all your information stored away to respond to infections that have previously been seen by the body. And the main distinguishing trait of this cell is you have a look at the nucleus, and the nucleus binds up basically the whole cytoplasm of the cell. So that's a major characteristic of that. You can't miss that. Here's another neutrophil. Here's another one over here. So essentially, this is what they look like. You can't go by them. There's another one. There's a platelet and lots of red blood cells. Okay, so we're going to move on to the second cell now, which is known as a lymphocyte. So here's a lymphocyte. Lymphocytes, they look very, very similar to another cell which is known as a monocyte. But the main thing that you want to look for in distinguishing between a monocyte and a lymphocyte is that lymphocytes don't have a curved nucleus, or don't typically contain a curved nucleus. I'll show you a lymphocyte soon, but essentially the lymphocyte usually has this curve in a nucleus and kind of stains a little bit differently. They're the same size, but lymphocytes tend to be obviously from the lymph fluid that you'll always find them within the lymphatic system, but sometimes you'll find them as a component of the blood. They're a very large cell, large nucleus, and they can be broken down into the natural killer cells, which are your inborn or innate cells that react to substances the body has already got systems in place to react to. But then they break down into T and B adaptive immune cells, which are your memory cells. So that's a lymphocyte. So when we compare it to a monocyte, we can see straight away that this distinctive curve within the nucleus is what kind of gives off a monocyte. Monocytes are about 10% of the white blood cells. Again, a single big nucleus. These guys form the innate immune system, or part of the innate immune system, and are one of the largest, or if not the largest, white blood cell within the blood. And interestingly, these guys, when they move out from the blood and they respond to an infection or inflammation, they actually can differentiate into a macrophage. So they've got an inborn macrophage function which allows them to gobble up things that are within that infected tissue that needs to be discarded through, of course, the process of endocytosis. So that's a monocyte. There's another T helper cell, or B helper cell. The next cell we're going to look at is, and excuse me if I get this wrong, because I always do, a eosinophil cell. Okay? Now, eosinophil cells make up about 3% of the blood. So you're lucky if you find them. They're not very prominent in the blood. But here's one here. Again, it's a little bit bigger than a red blood cell, as you can see. And as just for reference, when you're looking at red blood cells typically are about 8 micrometers in diameter. So they're a good reference not only to compare different cells within the blood, but when you're looking at histological stains of tissues and you want to measure kind of a rough estimate of a particular structure within a histological stain, you would kind of look at the red blood cell in reference to that tissue or component of that tissue, and you'd be able to kind of judge the size of it. But anyway, I'm mumbling on now. What we're more interested about is this eosinophil, or eosinophil. Eosinophilic means acid-loving, so they tend to be a little bit darker in their stain, as you can see here, compared to the neutrophil we saw before. So that's how they get their name. They're the second member of the polymorphonuclear family. So as you can see, if we zoom in a bit more without it getting too fuzzy, they have more than one component to the nucleus. So there's one here, smaller one here, and another one there. So when distinguishing between neutrophils, they essentially stain more because the acid stain that you used for a blood stain typically stains these guys a little bit harsher. And they also are kind of rarer to find. So if you do luckily see one, you'll be able to kind of determine it straight away because it will look different from a neutrophil. So always jump back to the neutrophil quickly if we can. Let's do that quickly. So we can see here the neutrophil. So it's a lighter stain. But when we jump back to the this cell, the eosinophil, we can see it's a darker stain. What else can I say? They usually combat parasitic infections or other infections with vertebrates, and they're heavily granulated. That's part of why they stain darker, not only because they're acid-loving cells, but they contain quite a bit of histamine and other immune proteins. The last cell we're going to look at is a very rare cell. You are very, very lucky if you find it. So here's a neutrophil, just while we're here. This is probably a dividing monocyte. Is a very rare cell, the basophil. So This is 2 here. They make up about 1% of the blood, so they're very, very rare. You'll be lucky to find them in a blood smear. But essentially, basophils you can't go by them. They're very, very heavily granulated, so they have a lot of histamine jam packed into the cell. So as you can see here, you can actually make out individual granules within the cell. So that's what kind of gives them away. And when you look at a basophil, the granules are so big and so numerous that they typically overshadow the nucleus that is kind of underlying the granules within this cell. They're the last member of the polymorphonuclear family. So I'll try to zoom in a little bit more on this stain, but they will have more than one component to the nucleus. So there's kind of an outline here and a kind of outline here and another one over here. Their main function is they're very, very similar to mast cells. So they're jam-packed of histamine, which is used in response to inflammation or to cause inflammation. But essentially you don't find mast cells within the blood. You find mast cells within connective tissues, and they respond to infections. But these guys will typically withdraw from the blood and essentially act like a mast cell, but they're only found in the blood, whilst mast cells are found within the connective tissues. So I think that's all I want to say. I'm running out of time. I've probably gone over time. So what I recommend you do to kind of learn how to look at a blood smear is you can use this site that I'm using, which is called, excuse me if I just move that over, these guys. So props to these guys. They have a whole library full of histological stains that you can look at in complementing your studies, and they're called Histology and Virtual Microscopy, and they're basically a resource at the University of Michigan Medical School. So they're very, very good to look at and complement your studies. So I highly recommend going to check these guys out. And full reference that all the slides that you've looked at today are from their website. So just quickly, red blood cells, no nucleus, donut shaped, lightly stained pink, platelets within our blood that form clots when respond to cuts and bruises and form clots to stop us from bleeding. Neutrophils, you can't go by them. They're usually found all over the blood, a type of white blood cell. The other cells we looked at are monocytes. Here's one here, a monocyte. Monocytes, neutrophils, lymphocytes, which look very similar to monocytes. Sorry, this is a lymphocyte, remember, because it doesn't have that peculiar curve within the nucleus. So this is actually a lymphocyte, whilst the monocyte, if I can find it just quickly, had the curve. There it is. The monocyte typically had the curve in the nucleus, and our T and B helper cells. And thanks for listening.